Hello everyone, my name is Abby and I'm going to be walking you through Lab Module 5 of the Yeast ORFan Gene Project. In this module, we will use various tools to help you predict where your gene's product is most likely to be found in the cell based on its sequence patterns. These tools include the Transmembrane, Helices hidden Markov models, SignalP, Psort2, and Phobius. So let's get started. Alright, and I'm going to open the worksheet and a guide from the Yeast ORFan Gene Project. And we're going to start out with the transmembrane helices hidden Markov models. And we're going to need to go to the SGD. I'm using the G&T MN2 today and get the protein FASTA sequence. And you're going to want to keep this up with you the rest of the module because we'll be going back and using it. So I'm going to paste it there, make sure extensive with graphics is selected, and submit. Alright, and this is what it looks like, and I am just going to take a screenshot of the graph and place it where it's asked for in the worksheet. Alright, and now we're looking for the number of predicted transmembrane helices, and that's 9, and the amino acid sequence range, and that's the 206.30992. And I'm going to copy and paste that as well. And now I'll go a bit into what the graph means and how to interpret it. So we have 9 bumps, if you count, which corresponds to the number of predicted transmembrane helices models. And we have transmembrane inside and outside showing. And if you can see with the blue line starting, we start inside, then we go outside, inside, outside, back and forth. And this thick blue line is also showing that we start inside the cell. So our N-terminus of the protein starts in the cell or in the lumen of the ER. It's starting inside the cell or inside the organelle. And so the 9 times it goes inside and outside corresponds to the number 9 predicted transmembrane helices model. So if that was a red thick line up there, it would show that it's outside and that the N-terminus starts outside of the cell or organelle. So now we're going to go and work with SignalP. So I'm going to open that. And go and get my FASTA sequence. And you want to make sure you have eukarya selected for organism, and then you would click submit. I'm going to change mine for the example proteins to use today. And wait for it to load. So this is what it looks like when it's loaded. And we're going to be looking for a SignalP score that is greater than 0.75 because that's significant. So this one right here would be significant based on that number. This one would be significant as well. And then this one would not be significant. And you can also see there's like no lines on the chart. So I'm going to work with this one. So I'm going to take a screenshot of it to paste into the Module 5 worksheet. And we're also looking for the signal peptide probabilities, which is 0.9813. And do these probabilities meet the cutoff? Yes, it's greater than 0.75. Alright, and now to go into how we interpret the graph. So down here we have our amino acids, and then we have these letters underneath denoting S, C, or the X. So the S corresponds to this red line up here. So a high S score indicates that corresponding amino acids are likely part of a signal peptide, like up here. And then down here it would have a low S score, which would signal a mature protein. And then we have this C, which would correspond to our cleavage site score, and there's a bunch of different green bumps. The green line also corresponds to the cleavage site, but the program has marked where the C appears as the corresponding amino acid as that being the cleavage site, and that also has the highest peak. And now we're going to start working with Psort. So Psort uses high throughput databases to find the physical location of your gene's product. So a couple things with this: it's all theoretical and based on computer programming. You can look at the manual for it and see that it really shows the basis for how they predict stuff. Another thing that you want to remember is that sometimes it will show that it looks like the protein is localizing to do 2 different spots of the cell, which it obviously can't. But again, this is all theoretical. And we also want to remember that protein localization can be impacted by conditions when we use this as well. All right, so we're going to want to go and get our FASTA sequence again and copy and paste that into the section for that. Make sure we have yeast animal selected and click submit. And this is what it'll look like when it loads. So we're going to get our PSG number, which is negative 4.40 for me, and the GVH, which is negative 3.66. So the PSG, a large positive score means there's a high possibility it possesses a signal sequence, but it's unrelated to the possibility of its cleavage. So in the manual for this, it doesn't really necessarily say what is a large positive score or what the numbers mean for anything. So I'm just going to say, since the PSG is negative, there is a low possibility it possesses a signal sequence. And then our GVH is the original weight matrix score for eukaryotes subtracted by 3.5. So a large output, there's a high possibility that it has a cleavable site. I also think that this is a low number. So I'm going to interpret this as ours likely doesn't have a cleavable sequence based on the GVH number. So the GVH seems that there is a low number and that it likely does not have a cleavable sequence. All right, and now we're looking for the ALOM score, which is negative 10.14. And it also asks for the number of transmembrane segments, so that's 9, so it crosses the membrane 9 times, and that lines up with our hidden Markov model prediction as well. Again, I'm not necessarily sure how to interpret the ALOM score. It doesn't really say in the manual, so for the interpretation, I'm just going to say it crosses the membrane 9 times. Alright, and now it's time to get our MIT-DISC score for the mitochondrial proteins. Which is negative 5.94. And the gavel, which says the cleavage site motif is not found. So MIT-DISC is a discriminant analysis to recognize mitochondrial targeting signals, and gavel is a consensus patterns around cleavage sites. Again, I'm not really sure how to interpret the MIT-DISC number, but since gavel says the cleavage site motif is not found, I'm going to interpret this as the protein is likely not going to be a mitochondrial protein. Alright, and now it's time to get the nuke disk information. And the score is negative 0.47. And that to me seems pretty low, so I'm going to interpret it as it's likely not a nuclear protein. And now onto the endoplasmic reticulum proteins. We're going to get the K-del information. And that says the ER retention motif in the C-terminus is none. And the K-del represents ER membrane retention signals. So for the specific ER membrane retention signals, we have this XXKK motif. So that is a dilysine motif. And it's a motif that exists near the C-termini of type 1a proteins. And alternatively, you could get the motif XXRR, which is a diarginine motif, and that exists near the C-terminus of type 2 proteins. So we have a dilysine motif that exists near the C-termini of type 1a proteins. Alright, and now we're on to peroxisomal proteins. And for SKL, so peroxisomal targeting signal in the C-terminus is none. And for SKL2, second peroxisomal targeting signal found. And then it has a short little amino acid sequence starting at the 112th amino acid. So I'm going to say it potentially could be a peroxisomal protein just because we found something for SKL2. But I'm not very certain or feel very strongly that it could be. So for VAC, the possible vacuolar targeting motif is none. So I'm going to interpret that as it's not a vacuolar protein. And lipid anchors for an MYR. And myristoylation pattern is none. The prenylation motif is also none. So I'm going to say for this one again that it's likely not a lipid anchor. So now on to the miscellaneous motifs, and the RNA binding motif is none, and the actin type actin binding motif for type 1 and for type 2 is none. For the MEMYORL, the transport motif from cell surface to the Golgi is none. The tyrosines in the tail is none. The dileucine motif in the tail is none. The DNA binding motifs is none as well. The ribosomal protein motifs is none also. And the NNCN prediction cytoplasmic reliability 94.1. So for my interpretation, it's not going to be any of these things, but it might be a cytoplasmic protein. So for the coiled coil structure, so coil is 0 residues, so it doesn't have a coiled structure. So overall Psort II summary and interpretation. So it looks like it potentially could be a peroxisomal protein. And then we have this information from the PSG and GVH. which I will include, and the fact that it is again saying from this information that it's crossing the membrane 9 times, and the information about the dilysine motif. So anything that stands out in terms of where a protein could be or where protein strongly could not be, you would want to include. So I'm going to say the protein crosses the membrane 9 times. So it could be a peroxisomal protein as well. And I'm going to copy and paste the dilysine motif information as well because I think that's important. And then the PSG and GBH information. And I should include as well that it predicted that it's a cytoplasmic protein. I just happened to miss that for this. So we're now going to get started with Phobius. So you'll want to open that and make sure that you do read through the guide for most of this because it is very important and helpful. So opening up Phobius and getting the FASTA sequence. And you want to make sure you have long with graphic selected, and then you can submit. And this is what it'll look like when it's loaded, and it's a very similar graph to the hidden Markov models. So you're going to want to screenshot that and get the Phobius data. I'm choosing not to get it, but if you scroll to the bottom, it's cut off, but there's a little thing that says Phobius data that's hyperlinked, and it'll take you there. It's a lot of information, so I didn't copy and paste it in. I would ask your professor if they want you to include that information. So interpreting the graph, it's very, very similar to the hidden Markov models graph. This one is a little more sensitive, so you might have it crossing the membrane more than 9 times, or how many times it originally showed up, but mine is still crossing the membrane 9 times. And it's a little easier to read as well, as it states it as cytoplasmic versus non-cytoplasmic. So mine starts off with that blue as non-cytoplasmic, so the N-terminus is going to be either like in the cell or in the lumen of an organelle. So my hypothesis is my protein crosses the membrane 9 times because it didn't pick up on anything else in this second model, with the N-terminus sticking into the lumen or being non-cytoplasmic. It potentially could be a peroxisomal protein, but as of right now, we're not sure, and this is based solely on the PSORTII data. And it does not strongly support point to it being a peroxisomal protein. And so that's it for this module. And you might want to look into everything a little bit more in depth or follow up with wet lab experiments. Thank you for watching. I hope that this video was helpful. For more information, go to the yeastorfanproject.com, and this video was funded in part by the National Science Foundation. Thanks!